we're all set. Well, welcome everyone. It's great to see familiar and new faces. My name is Elisa Atkins. I'm your representative of 10x Genomics. We're super excited to be here today to launch the spatial biology core with Sarah Mazzilli and Roxy. I don't know if Roxy yet, just running an experiment.
Yes, so she'll be right down. She's working with Shonia and others.
I will let Sarah and Roxy introduce themselves, but I just want to really quickly say that 10x Spatial group is here. Yours truly, Jeff Weiland, is your Senior Science and Technology Advisor with 10x Genomics. Shonia Rose in the back is your Field Application Scientist. And so we're super excited to bring Visium HD to BU community. Welcome in, please grab some lunch and find a few seats out front here. And so we're running this promotion to really get people started. It's 50% off up to a few kids with Visium HD. Please talk to Sarah whether you want to do probe-based approach, human or mouse, or if you're working on any other species. We have this new kit that's Visium HD 3 Prime that you'll be able to analyze any species. Or you can even do long-read sequencing and discover TCR, BCR. And so, Sarah, please take it away. I really wanted to bring in a red ribbon and do like a cutting ceremony, but we all can imagine that it's really, really exciting. So, welcome, Sarah.
Thank you. So thank you, everyone. It's great to have a nice full room. This hopefully, shows the excitement around spatial biology, and I think this has been moving forward. You see our famous Ruben Dries in the back, who really pushed, I think, us into a spatial biology generation here at BU. And we're going to hope to expand that and get other people excited and help you generate some data. So first and foremost, as Alyssa said, and I can't say enough good about 10x. They've really been a partner in this process, and then Yuri could speak to them as well in the single cell space. Been working closely with them for many years now to get our spatial technology up and going, but also in the single cell. So I encourage you to keep working with them. Their customer service is outstanding, and they'll keep helping us and helping you as an individual user. But who are we here at BU? I'm an assistant professor in the section of computational biomedicine, where my background is really in molecular biology. I joined the section to work with Avi Spear as a postdoc to gain experience in computational biology, gain experience in next generation sequencing at the time, and really have grown kind of out of standard bulk profiling approaches and thinking more in the spatial and single cell space. And so I joined BU 12 years ago and have been on faculty for about 7 years. And as part of that, had Roxana join my lab as a postdoc, where she has recently been promoted out of her postdoc role to a senior scientist in our group. And she'll be helping lead kind of this initiative as we kind of get going, and we hope to expand in the future. But between the two of us, we're hoping to help you navigate your journey. And so what technologies are we using? Get that out of the way. Is we have two technologies. We're going to hear today about the Visium technology, and so I won't talk too much about it. I'll let Jeff give you the kind of full rundown of how the instrument works. But this is the tool that we're using to do spatial transcriptomics, although there are paths to not using this instrument for the Visium assays, and we're happy to talk about that. That's kind of the older generation of technology. But most of the new assays that are Visium specific, so that's all of the whole transcriptome, spatial transcriptomics. Jeff may also talk about the Xenium technology that our collaborators over on the Charles River campus have, and we're working on a pathway to working with them as well. In addition, we have a proteomic instrument, and so this is highly multiplexed. We can multiplex up to 40 or 50 proteins together and they're bladed in a spatial way. And so you see kind of an example of an airway biopsy there. And our ability to localize all kinds of populations, much like single cell and spatial transcriptomic technology has allowed us. And so we're excited to talk about that. We'll bring in standard bio tools again to be able to talk more about that and give you kind of a similar rundown. And so, what is the spatial biology core going to do? I think that's been a big question. And so not only are we going to give you opportunity to generate these types of data, but we're also hoping to help you navigate working in the tissue space. And so we work closely, and I'll show on the next slide, who but it's important to process your samples, whether they're frozen, whether they're fixed, and how you're generating those for your spatial technologies will work with you, will help you connect with cores here at BU that will enable you to process your samples properly, and talk you through how you collect those samples in animal models or in human studies to be able to do these types of assays. And then we'll work closely with Yuri's group for the sequencing-based technologies and in order to sequence your samples and help you understand and navigate that process. And then working closely with some of the analytical groups here, even within the spatial core and as part of the single-cell core on the analytical front. And so who are those cores? And so as I mentioned, 2 pathology cores, one with Eric Burks, who primarily processes clinical samples and has kind of standard workflows and may help you work on if you have blocks from an outside collaborator or from an internal collaborator that you need to properly section. They also have lots of different kind of staining technologies and slide scanning, as well as working with Nick Crossland's group in the Needle, where they have a lot of technology that will span basically any species, any disease model to help process your samples for these technologies. And then similarly, as I mentioned, working both with the single cell and microarray and sequencing cores through Yuri and Josh to be able to both generate your data, but also on the analytical side. And so these are kind of a list of the different services. Again, on the left-hand side, that's that IMC or image mass cytometry and some of the workflows. And again, we'll have a separate seminar to talk specifically about those workflows that we'll kind of advertise to the group that's here today in addition to the wider BU audience. As well as, so for the 10x technology, this will be anything from working with you to QC your tissues, make sure that they're amenable to the spatial technologies, working with you on your imaging approach that goes in companionship with the Visium run, and then ultimately giving you a path to using the Visium instrument yourself. You're a postdoc in your lab that's gung-ho, you're going to process 20 samples, they'd really like to do the process. We're going to enable that. But we're also very happy to just take your samples and help you, get them through the pipeline and generate your data, and again, working collaboratively with Yuri's group in order to do that. We are having a lot of questions about Xenium. We're hopeful in the future that we'll continue to grow. So use our core so that we can show to CBN and the Department of Medicine that this is exciting. People are wanting this technology, and it gives us an opportunity to figure out how to grow and possibly bring a Xenium to this campus in addition to working collaboratively with Jerry Chen's group. So just a quick workflow. How is this all going to work? We're going to meet with you. So I'll show you our new email address on the next slide. We'll kind of help you guide and think about your experiment before you start coming with us with a tissue section. We'll talk about the different needs of the core, and then ultimately help you make sure your samples are appropriate, talk to you about what area in your tissue sample that you're gonna ultimately use for analysis, and then talk you through kind of the different places. And like I have up there a little bubble, we'll also, this is the opportunity for, to train different folks, and then we'll walk you through the different steps as we get through it. So that's kind of it. I'll flash this slide up here again. As Lisa said that we are running this special. We hope to continue to collaborate with 10x and think of additional specials, but I want you to really note the time. We're trying to get new investigators in the short term, and that doesn't mean that you need to have samples by the September 26th to be able to process. This means that it gives you you have up to 6 months usually to use most of the reagents, and so we can be thinking in the early part of later this year or early next year. So if you have tissue samples kind of in that vein, or you think you're going to be able to generate that data, we're happy to work with you. There is going to be, so these are costs right here for the actual reagents. There is a cost of running it. It's going to be about $200 a reaction if you do it yourself, or it's going to be about $400 a reaction if you have us do it. And so that's the workflow of generating the library, et cetera, and the additional reagents outside of the standard kit that are needed to be able to process those samples. And so that's just for this part of the component, and there will be a few other small fees associated with imaging, et cetera, and we're happy to explain that to you when we get there.
I just want to add that outside of the whole kind of greater Boston area, these fees are probably the lowest I've ever seen, so I really hope the BU community takes advantage before the core is so busy that these group rates the fees.
Yeah, so we're really trying to make it accessible, and we may find out we're undercharging, but hopefully this is the charge that we calculated to be what we need to be able to get people in the door and be able to do this, and we'll work with you to try to figure out how to, you know, work with 10x to get the appropriate reagents and be able to run your samples, particularly pilot studies, as you know, the kits will come standard in either 4 or 16 reactions, for example, and we'll help you navigate either how to get just a single slide to generate pilot data in order to start to be able to have a grant that would support a larger study. And so with that, I'm going to turn it over to Jeff. He's going to walk you through some of the Visium technologies and maybe give you some ideas of the different analytical approaches that others are taking, that 10x enables, and I'm sure much more. But we do have a new email address, so as you know, you can certainly still email me and Roxana. But this is going to be our email address for the Spatial Core, and so we look forward to getting your emails. But certainly, if you are interested in taking advantage of the deal, try to reach out to us in the next day or two so we can work closely with Elisa to be able to generate those quotes and get your orders in on time. All right. Any questions from anybody? All right. So with that, I'll turn it over to you, Jeff.
All right. Thank you.
You're sitting next to an empty chair. Can you just raise your hand so folks can? Or will chairs here and these people want them? Oh, okay.
All right, let's get this up and going. Great. Okay. Well, thank you, Sarah, and congrats on the progress. It's exciting to be at this point for the core, and I think for this technology here at BU. I'm going to take a minute to give an overview of the technology that's here, the Visium technology, and try to give you some context for that technology, and then time allowing, we'll go into talking a little bit about data analysis. There's a lot of talk about getting data and using the assay and how it works, but it's worth thinking a little bit about once you have the data, what do you do with it, right? And so we'll go into that as well. Just by way of introduction, it may not be necessary for most of you, but at 10x we develop tools that grapple with biological complexity. These include our Chromium technology. This is something that 10x came out with over a decade ago, and allows you to take a piece of tissue, break it down into individual cells, and then cell by cell look at the molecular contents of that cell. And as that assay has grown and been developed over time, 10x has spent a significant amount of time in research and development. Then the types of molecules you can look at in those cells have gone from just looking at 3' gene expression to 5', looking at proteins, looking at chromatin states, doing any one of those individually or all of them together, right? So you can look at most molecules in a cell on a cell-by-cell basis. We've also expanded our capabilities to scale the assay, in addition to coming out with new approaches on the Chromium instrument. We've recently acquired a company called Scale that has some really interesting technology for plate-based single-cell transcriptomics that we're integrating. And so if that's of interest to you, whether you're working with solid tissues that need to be dissociated or with liquid tissues that are already dissociated, then please reach out, because it's an amazing technology. But I'll be primarily focusing on our additional technologies that we've come out with more recently, and primarily Visium. But we also have, as Sarah mentioned, our Xenium in situ platform. Both of these were developed with the goal of providing additional context to the data that you can generate with single-cell technology, right? Most of us exist today because our cells exist in space, right? There are complex arrangements of cells into tissues and organs and organisms, right? And so being able to look at that context, not just an isolated cell with its molecules, but in the context of a tissue, it adds a valuable level of complexity that now we can interrogate. And the reason we have two platforms is that each addresses this spatial question from a different angle and can be applied differently. And just to give you context for how these sit in the state of the art, I really like this diagram from a review article that came out recently. And what we're doing here is we're essentially trying to solve the problem of how do you do multiplexed imaging, right? How do you take and combine molecular data with an image and do it on a large scale so that we're looking at not just a few markers like we do with immunofluorescence or IHC, or 1 or 2 genes like we do with in situ, but we're looking at hundreds to thousands of genes across millions of transcript targets, for instance, and then putting that in an imaging context. And in the field, there are a couple of approaches that have kind of become dominant in terms of how they're being deployed, both academically but primarily commercially, because of their consistency. And I've set this up so that we essentially have 2 columns. One I've labeled imaging methods, the other I've labeled sequencing methods. And in the imaging methods category, we have in situ hybridization and in situ sequencing. These approaches, they use some kind of an imaging approach to directly visualize in tissue the location of the target molecule. In this case, we're talking about a transcript, right, an RNA transcript. I love it. I wish I had a soundtrack every time. And so what we're looking at are RNA transcripts in the tissue. And with these approaches, we directly label those RNA transcripts, and then we observe them using an imaging system. And there are a couple of approaches, right? So in situ hybridization is where we add some kind of a probe, and then we introduce fluorescently tagged conjugates that bind to those probes, and then we observe those with a fluorescence microscope. Typically, we're doing this in a multiplexed fashion, so we would remove that first round, we'd add another round, and then image again, and then by doing this in a cyclical fashion, then we can observe across thousands of genes and millions of transcripts in a single piece of tissue using a standard fluorescence microscopy approach. In situ sequencing is similar, where we're directly labeling transcripts with some kind of a probe, adding conjugated fluorophores, and then imaging. But the difference here is that we can get more information from the transcripts beyond where are they located, what's their identity, and some level of quantification. We can actually start to look at the sequence of the transcript. So we can start to understand what base is where, and we can look at things like isoforms, single nucleotide variants, things like this. So there are various levels of in situ sequencing approaches. So this is on the imaging-based side. On this side, I'm calling these sequencing-based methods. And there are a couple of approaches for using the sequencing-based methods, but essentially what it boils down to is adding some kind of a tag to the transcripts that you're looking at, and then when you sequence, you can see that tag. And that tag corresponds to an XY coordinate system. And so you can translate that coordinate tag to a location after you've done the sequencing, and this is how these approaches work. And the 2 most prominent approaches, although by far most approaches now are in this array category, include this array-based approach where you take your tissue and an array is either superimposed on the tissue or the tissue is placed on an array, and then some kind of a tag is either added to the tissue from the array, or the molecules in the tissue are liberated out of the tissue onto the array and captured. And then once captured, then you add that spatial tag. This approach, called microdissection, is a little bit cumbersome and not quite as common anymore. But essentially what you do is you label your targets of interest with some kind of a fluorophore, and you add a conjugate that allows you to then detect and capture it. And so you'll liberate that using microdissection. The microdissection portions are put into some kind of a plate, and then they're imaged, then they're analyzed in a sequencing system. This approach, the resolution is limited and the sensitivity can be affected, and so it's really kind of relegated to niche applications these days. Most of the approaches, like I said, are these sequencing-based approaches. And so this is where our platforms fit in terms of the state of the art right now. So we're developing and offering the Xenium platform as an in situ sequencing approach. I put it in this category because we can use it to look at a little bit the sequence of the transcripts that we're working with, and not just their identity and location, although that is also included. And then the Visium is our sequencing-based approach, which I'll be spending most of my time talking about today. The reason that we're developing both of these in parallel is that when it comes to looking at the entire transcriptome or doing discovery-based spatial transcriptomics, then these sequencing-based approaches are usually the best right now. The state of the art really doesn't support using these in situ approaches yet for looking at the whole transcriptome, just because of the way the technology works. You can kind of force the issue and try, and I know some are, but the results are typically inconsistent and not up to the quality that most of us are looking for. And so in the meantime, these sequencing-based methods really shine. Also, because of the data set you get, it's a sequencing data set, right? So you can use that like you would any other sequencing data, and it becomes, I guess it sounds a little cliche, but it's really a gold mine of information, and then it's a matter of figuring out how to mine it, right, to get the most data out, or most value out of it. But that's why we're pursuing both of these. And so if we look at these side by side and talk a little bit about why we have both of these and why you might consider using one or the other, it kind of boils down to unbiased discovery versus precision insights, right? So the Visium assay is designed and supported as a whole transcriptome discovery, spatial discovery approach. It's compatible with H&E and immunofluorescence. I've already mentioned it's sequencing based. We're at about the single cell scale right now. So we're collecting data at a 2-micron resolution with the Visium platform, and I'll show you how that works a little bit later. And then we have a high transcript diversity, right? We're looking across the whole transcriptome. With Xenium, we're focusing on a subset of the transcriptome. We're up to just over 5,000 transcripts that we can assay for at a time. We can still do H&E and IF with this assay. It's high resolution imaging based, so we're using microscopy techniques that allow us to get a resolution of about 30 nanometers in X and Y. So we're doing molecular detection, right? So we can actually detect the location of transcripts within cells. And then we're actually also localizing the Z position of each transcript. And this is something that's often forgotten, but each Xenium data set has the Z position as well. And just because of the nature of optics, that resolution is just over 100 nanometers, right? So we get X, Y, and Z on the nanometer scale for every transcript that's detected in the tissue. And of course, that means it's subcellular, and we have a very high per gene sensitivity. We're focusing on a subset of the transcriptome, but we're very sensitive when it comes to counting individual copies of each transcript. And so this is one of the advantages of this approach. Okay, so this is Visium and Xenium on a high level. Now I'm going to dive into Visium specifically and just talk a little bit about how it works and introduce you to some of the new stuff that's come out. So first, and I think this number is already outdated since I put it up last, but we're looking at close to 1,000 publications on the Visium platform alone. And this has been growing rapidly since it first came out. This is a platform, the goal here isn't to brag necessarily, but it's to point out that this is a platform that is a known quantity now. It's not something that needs to be proven so much as something that people are looking for and expecting in a lot of these studies now, especially when you're doing molecular studies using tissues. And so, this adoption shows that it's a very compelling technology, but also that it's a technology that researchers are relying on to get high quality data. And so, all right, there are a lot of people using it, but what's so compelling about this technology? And for those of you who haven't seen it much or just sort of starting to think about this, the reason this technology is so compelling is it allows us to generate a couple of types of data. One of those is an H&E image, and this is something that, or an IF, but these imaging data are something that we've relied on in scientific research for a long time. And especially something like an H&E stain, it's fairly simple and straightforward, but it gives us a lot of information, right? We can look at this piece of data right here, and we can extract information just looking at it, right? We can see areas where we have these, distinct mucosal structures. We can see this tumor. We can see infiltration of immune cells. We can see some granulation tissue, right? So already just looking at this, there's a lot of information. But then if we employ modern digital histology approaches, we can extract quantitative data from this, right? We can see how many cells are present. We can start to look at the progression of the disease. We can start to grade it, right? So there's a lot of information you can extract from this. But that's not all the information this tissue has for us. There's more information here, and that's what's on the molecular level. And specifically, we're talking about the transcriptome. And of course, being able to look at the transcriptome in the context of this tissue allows us then to get a better idea of what's happening in this tissue in its complex environment. And so that's what the Visium assay does, is it pairs gene expression data with the histological data to give us a full and complete data set so that we can understand what's happening in this tissue in its complexity. And so what you're looking at here on the right-hand side now is an example of a Visium data output from the same exact tissue section that we collected the H&E image from. And this data now represents gene expression profiles. So each color is a gene expression profile that's been determined in an unbiased fashion. So we call these gene expression clusters, a little bit different from the single cell data, which are cell clusters, right? We can say this is a cell type, this is a cell type. Here we're looking at gene expression, and each different color represents a different gene expression pattern. So what you can see clearly is that these gene expression patterns line up nicely with some of the features that I pointed out on the left, right? And allow us now to get molecular characterization of the morphological information that we were able to extract. So this is what Visium does, essentially, right? Kind of from a simple overview perspective. And the way that this is done has changed a little bit over the years. We've introduced some new approaches, and recently we have come out with a technology called Visium HD. And Visium HD allows us to collect at 2 micron resolution, high quality whole transcriptome data. And there are a couple of ways to do this. One is with what we call our Visium HD whole transcriptome panel gene expression approach. And this covers the whole protein coding transcriptome. And the way it works is that we have probe sets that are designed to hybridize to target RNA, and then those probes are essentially what we end up capturing, tagging, and sequencing in our assay. And the reason this approach works so well is that these probes are relatively small, and they target about 40 base pair chunks of RNA, and so they work in a variety of sample quality types and preparation approaches, and they do it very consistently, and they're also much more efficient to tag and to sequence. And so this approach is very good, very reliable for FFPE tissues, for example, or fresh frozen tissues that may have somewhat of a lower quality, and even fixed frozen tissues, right? So this is a very good approach for doing that. It's very consistent, and in cases where you're,
I just want to ask the room, how many of you are working with fresh tissues? Higher, higher. Okay, good amount. What about fixed or FFPE? Okay, a little bit more. What about human? Okay, what about mouse? A little bit more mouse. What about non-human, non-mouse?
There you go. One brave soul. Very nice.
Well, thank you for sharing. It's a good mix.
Yeah, it looks like we've got a broad array of interests here, which is good. And so with this whole transcriptome approach, it's excellent for determining which transcripts you have, where they are, and quantifying them efficiently. And in fact, this is the assay that we recommend for most people. There are a couple of caveats to this assay, though, version of the assay. One is that we have these whole transcriptome probe sets that we've designed. They work very well and efficiently, and they're targeted at human and mouse. So for those of you who aren't looking at human or mouse, then you can either make your own probe set, which can be time-consuming and expensive, or you can use our other approach. The other thing is that this is, of course, designed to look at protein coding RNA, right? So if you have situations where you're looking at other types of RNA, then again, you may want to go to this other assay on the right that I'll tell you about. Or if your questions are around things like isoforms, SNVs, differ' kinds of variants of transcripts, then again, you may want to move over to the other assay that we've recently released. And this is our Visium HD 3' gene expression assay. This is an adaptation of our original version 1 chemistry, where we were placing tissues directly on a slide, and capturing transcripts at a resolution of about 55 microns to the HD resolution and the site assist. And so with this technology, we're actually directly capturing by the 3' end transcripts from the tissue. And so with this approach, we are getting the most diverse transcript capture that you can get at a high resolution assay. And it's species agnostic, right? If your species expresses RNA with a poly A on it, then we can grab it. And even if it doesn't, you can add the poly A and grab it, right? There's approaches for doing this, and I can go into that, on a one-on-one basis. And so with this approach now, we can grab whole transcripts from the tissues. And so if you need one of these types of applications, then this assay is available now for your use. And we're happy to consult with you. I know that the folks at the core are also happy to consult with you in terms of which one would be better for your experiment. Okay, so these are the types of Visium assay that we're offering, we think that this is a pretty comprehensive offering for now in terms of being able to look at transcripts across the whole transcriptome in a tissue. Now that you've seen it, then some of you might be thinking, well, how is this done? For those of you that are, then that's great. And those of you who weren't, hopefully now you are thinking that. Because this is how the assay is done. And I bring this up just so that as you consider doing this assay and think about moving into it, you can have in your mind a concept of what needs to be done for preparation. But essentially, what we're doing with the Visium assay is we're bringing together two workflows. One is a traditional histology workflow where you're sectioning tissue onto a slide, you're staining that tissue, and then you're imaging that tissue to collect a scan, like a whole tissue scan. So that's on this half of the assay here, right? And then on the other half, we are doing a sequencing run, right? This is just next-gen sequencing. And we bring both of these together via this device called the site assist. And the way that this works is that after you've sectioned, placed, stained, and imaged your tissue like you would with histology, then you'll move into the RNA isolation portion. If you're using the whole transcriptome version, then you'll incubate with the probes and then move on to the site assist instrument. If you're not, then you'll just go directly to capture of transcripts on the site assist device. And so the way that this site assist device works is that we can take tissue and place it into the instrument. So here we have tissue, we can do 2 slides at a time, and we place this into the lid of the device, and we align it with the lid, right? There are some alignment guides that we have that help you to do this. And so you align your tissue so that it's lined up with these capture windows, and then you take a Visium spatial gene expression slide and you place it into the base of the device. And then what we do is we close the lid. Once the lid is closed, then this facilitates the overlap of the tissue with the Visium slide. And on the Visium slide is an array, kind of like what I showed earlier, right? And this array is set up with capture oligo sequences. And so what we do is inside the site assist device, once the tissue on the slide is made, contact with the Visium spatial gene expression slide, then we liberate the molecular contents out of the tissue onto the Visium spatial gene expression slide, where they're captured by these oligo capture sequences. If you're using the whole transcriptome probe-based approach, then it's the probes that are coming out and being captured on the slide. If you're using the 3' approach, then we're directly capturing transcripts by their polyadenylation sequence on these Visium slides. Once that transfer process is complete, then we open the lid back up, we pull the Visium spatial gene expression slides with those array capture areas out, and then we can proceed with processing on the back end. At this point, the tissue has essentially been digested and is typically not suitable for any other applications. But we proceed then with the Visium slide. And so then that's where we get to the back half, which is now where we do reverse transcription. We add the spatial barcode to each captured transcript or probe. And then for each capture area, we generate a library. So we have a tube where we elute the captured and tagged molecules off of the slide into a tube, and then we use that to generate a library, and then we sequence that library. So, pretty straightforward. The techniques that we use on the front end are all standard histology techniques. The techniques we use on the back end are all standard sequencing techniques. And then we provide the protocols and the reagents and the instrumentation to kind of bring these two together. And I think it's very approachable for anyone who's thinking of trying it. Now, one thing I'll show you is, the Visium spatial gene expression slide is kind of the key to this technology. And so I'll show you how this works. So these slides are designed so that we have 2 capture areas per slide. And each capture area, like I said, is essentially an array. And what this array looks like is it's made up of these 2 micron squares. And each one of these 2 micron squares is in turn made up of millions of oligonucleotide capture sequences that are conjugated to the glass. And if we were to kind of zoom in and then zoom in again, this is what one of those oligo capture sequences looks like. This end is conjugated to the glass, and then this end is what's pointing up towards the tissue. And so when we liberate the molecular contents, anything with a polyamylation sequence will be captured by this poly-DT. And then after capture, then we add a spatial barcode, a unique molecular identifier, and then this sequencing tag. And then we pull this off. So you'll note a couple of things. One is that the smallest unit of resolution here is 2 microns. So we're collecting at, for the average cell, subcellular level, right? But that you can sum up multiple of these 2 micron squares into larger features called bins. This is a little bit like what we can do in microscopy, where we can take multiple pixels and we can sum up their signal to create a larger feature and bin it. And there are a few reasons why you'd want to do this, right? One is that you may want to enhance the signal of your output, right? So this is a great way to do that, because now you're summing up the signal from everything in this bin. The other is to facilitate a more efficient analysis, right? It's a lot easier to analyze larger units that there are fewer of than smaller units that there are more of, right? The other, and I think this is the most important, is that you might want to match the biological features that you're trying to study. So, if you're thinking about a cell, which is what a lot of us are trying to study, they don't tend to express transcripts in discrete 2 micron squares, nor are they regularly arranged, right, in the tissue. And so by creating these bins, we can more accurately depict the cells that we're trying to study and match the expression to those units, right? And there are some ways to do this. One is the example I've shown here, where we're just creating kind of a generic square. But the other approach is to do something called binning-based segmentation-based binning. And the way that this works is that we take advantage of the fact that we have an image. And that image typically has a cellular level feature like a nucleus that we can use to create a custom bin. And so the way this works is illustrated here. So represented in this illustration are cells. So that's what these borders represent, as a cell wall, or a cell membrane, excuse me. Although with our new 3' you could be looking at plants. But this is a cell membrane. And then we also have a nucleus here. And then the kind of graph paper looking thing here represents that Visium grid array. And they're both overlaid. And so one approach we can take is we can create kind of just generic 8 micron square bins. And this is fine. This actually generates some pretty nice data and is one of the default outputs from the Visium software that we provide. And it gives you good regional expression that generally aligns with cells. But if we look closely, then we can start to see that if we really want cellular level data, we're getting a little bit of mixing going on, right? So if we call this bin a cell, then we're getting most of it, but some of that is spilling over into this bin, and now this bin is also clipping this cell, right? And so what we're getting is kind of an average of the cell to the left and the cell to the right, and we're calling this bin another cell, maybe, right? And you can see this kind of goes on, right? So if you want to get cellular level information, this is a little bit inaccurate, right? So what if instead you create a bin that is based on the shape of the nucleus. And this is something we can do. So we can do something called segmentation, where we identify the area in the image that contains a feature like a nucleus. And then we match that with the XY coordinates of the transcription data. And then we can create a new feature that is shaped a bit more like a nucleus. So now we get the gene expression under this array area here. And then we can actually take that and we can expand it out a set amount, right? A set distance or a set number of these 2 micron capture squares. And then we can approximate a cell much more accurately. And that's what we're looking at here. Now this approach is really good because it allows you then to get some cellular level data and can kind of augment this type of an approach. One thing that you'll find though is that if there's no good staining of the nucleus, or for whatever reason, maybe it's out of the plane, maybe it's out of the picture, maybe the stain didn't take, then you're going to miss those data, right? And so it's not the perfect way to go, but it's another tool in your toolbox to ask certain questions of the data that you couldn't ask before. And so by using both of these approaches, you can get a really nice data set and answer different types of questions. So what does that look like in real life? That was a diagram. So we now kind of conceptually understand what's going on. Let's look at this applied to real tissue. So here we're looking at intestinal tissue. We're looking at something along the top called a Pire patch that includes a lot of immune infiltration into the tissue. And you can see that the H&E image on the left clearly indicates the morphology that's characteristic of this type of structure in the tissue. We have a normal below so that we can get some comparison. In the center, we're doing that 8 micron general clustering that I described. And again, the colors represent gene expression profiles that have been determined based on the expression data, which genes are up, down, which ones are associated with each other, and in space. And so we can see these two examples give us a good indication of what's happening kind of at the regional level, right? So, you know, we get good alignment between different cellular niches, for example, and regions, and maybe some cell types that are present. But when we do the segmentation-based clustering, where now we're using those nuclei to identify where cells are, and then reclustering, we get a lot more heterogeneity represented in the cell populations. And so now we're going beyond just kind of niche analysis, and we're getting cell types, even cell types, subtypes, cell states that start to come out, right? So the same type of cell, you might have immune cells that are activated, deactivated, exhausted, you know, there's different types, right? And so now we can start to see that a little bit better when we do this approach. Again, you can see too that you know, there are areas where we may get a little bit of dropout, which we didn't get over here, but we're getting a more nuanced look at the cellular populations, right? And so these are how you might use this approach. Another example is shown here. This is looking at human ovary, and we're looking at carcinoma. And again, we can look at this segmentation-based approach. And here we're using the nucleus detection plus some expansion to identify individual cells in the tissue. And doing this, we can get a really nice high-resolution look. And by high resolution, I mean, of course, spatial resolution, right? But also cellular resolution, right? The number of types of cells that we can see, distinct cell types, to the point that we can actually look at the tumor markers here and the macrophage markers, and we can get a really good idea of what's happening in the immune space and the tumor space in this tumor microenvironment using this approach, and we can see those relationships in space. Yeah?
Just want to add that what's really exciting is that it's a software update, and so folks who have done this EMHD previously now can go back and reanalyze the data with this new update with Space Ranger and the Root browser, and based on the feedback from the customers so far, that they are seeing a lot better differentiation between the different cell subtypes, fibrotic tissue that are really hard to differentiate previously. So it's a lot of positive feedback. So yeah, if you have tried to either if you already have the data, please let us know how it goes. If you have reanalyzed it, or if you're just going to data analysis. Sorry, there's a question here.
How many genes do you get from each nucleus?
How many genes?
Yeah.
So we're surveying the whole transcriptome, right? But of course, each nucleus isn't expressing every gene. So it depends on the tissue type and which genes are being expressed. But we're usually looking at thousands of genes that we can see in each cell.
Because single cell sequencing doesn't allow to detect like whole transcriptome, right? It's only 15-20% of the top genes. Do you have the same situation here, or it's like really full transcriptome?
Yeah, that's essentially true, right? So you're going to get a similar level of sensitivity with this assay, maybe a little bit less than what you're going to get with the single cell RNA sequencing for this. Yeah.
So practically you will see only like, probably housekeeping genes, right? Very highly expressed genes, but not really like, regulatory genes like transcription factors.
I don't think that's true in this assay, right? So, and even in the data that I'm showing here, right, these aren't just regulatory or housekeeping genes that I'm showing. So I don't think that that's an accurate way to describe the type of data that you're getting. You can get some very nuanced data. You can get nuanced data from single cell as well, right? So, you will see high expressors for sure. And oftentimes the high expressors will take most of the sequencing, space. But you can also get low expressors. There's quite a variety. I would say the limit of detection, it varies, but I think it's low enough to allow you to see a variety of informative genes in each one of these areas that we would call a cell.
I'd also add to that, like Jeff is alluding to, the quality of the input really plays a huge factor to what you can get out, and your sequencing depth can vary, so you can try to get to things like rare transcripts by sequencing a little bit deeper, if that would be beneficial.
Yeah, that's right. So on the sequencing side, there's a point of saturation, of course, but before you hit that, then you can increase your sequencing depth to generate more sensitivity from the data. And so if you have a high quality input sample and you have a good sequencing depth, I think you'll find that you're actually getting a very nuanced picture of what the gene expression looks like. Now, the good news too is that these assays are designed to work with a variety of tissue types without the need for extensive optimization. We have a universally optimized protocol that we offer that works across most tissue types. We also have, for various tissue types, preparation guidance for how to prepare the tissue in a high quality fashion before it even goes on the slide. These are some examples here of tissues that have been successfully used, specifically with the cell segmentation. This is a very versatile approach, and at 10x we do a pretty good job, I think, of creating assays that are broadly useful, right, and broadly accurate. And I think you'll find that that's the case in most applications. But we also understand that for some, there's been a lot of work that's put into things like segmentation already, right? So if you've done your own segmentation pipelines, you're using different segmentation software, then we have options available for you to do this type of analysis on your own and then plug that back into our Space Ranger software and then analyze the data that way. You can adjust the settings of the Space Ranger segmentation pipeline. So there are a lot of ways for you to interact with this approach that allow for a lot of customization if you don't want to use the turnkey segmentation that's built into the Space Ranger software. So there's a lot of flexibility. I think the takeaway here is that come talk to us if you have questions about segmentation, combining expression and image analysis. There are a lot of really nice options available now. And then finally, I'm going to give you a little bit of a sneak preview. So I've talked a lot about what is available today, and that's the result of a lot of hard work and a lot of development. And if I gave this talk even 8 months ago, some of this wouldn't have been in here. So this is moving very quickly. But we're really excited to announce that we'll be releasing soon a Visium HD XL, we're calling it, where you can look at a lot more tissue in these capture areas. Right now, a Visium HD capture area is 6.5 by 6.5 millimeters. And so, whatever you can fit in there, you can analyze. But we're going to be increasing the size to 11 by 11 so that you can either multiplex tissues much more easily by putting many pieces in one area, or looking at much larger pieces of tissue, which I think is an exciting development.
Can we see a show of hands here too of people who are working with really large tissues who thinks this might be beneficial? This is about 11 by 11. Okay, good people, great.
Good. So for you, then you can look forward to this coming out soon. But like I said, it's also a great way to take advantage of the space by multiplexing smaller tissues, right, where you can put multiple in the same capture area. And so, this is coming soon. I don't have a specific release date on this yet, but it's something to think about as you're planning experiments that we're going to be offering this. Okay. So how much time do I have left? About 5 minutes or so?
Yeah, about 5 minutes. I'm going to take a couple minutes at the end. A couple questions.
Okay. Yeah, so I think then what I'll do is I'll say this. When we are generating data, it's important to think a little bit about what those data look like. So after the assay is complete, on the bench side, I guess, then you're going to have two outputs, right? You're going to have an image that you collected of the tissue, so a tissue scan, and then you're going to have sequencing data. And these two data outputs are used to generate the figures that I've shown you so far. The first step of your journey for analysis is going to be to use our Space Ranger software. And Space Ranger software is something we provide. It's free of charge, and it's designed to run in a Linux environment, typically on a computing cluster, and runs on commands. So this is a command-based pipeline, or set of pipelines. And the way that this works is that it's used to essentially process the image and the sequencing data and align the sequencing and the imaging data so that you have spatially resolved transcription data that is aligned to an image. So that happens in Space Ranger. Once that's completed, then we move into our Loop Browser software. And in Loop Browser, we can visualize the data. And Loop Browser is designed to run on a personal computer, so a Windows or a Mac computer. And with this, you can visually interact with your data. You can look at the spatial arrangement of the tissues. And this data, or this package, is again free. You can download it from the internet. And it's designed to be kind of a turnkey solution. You can do a lot with the Loop Browser. You can generate figures. You can look at distribution of gene expression. You can do differential gene expression analysis. You can create custom feature lists and look at their expression across the tissue. You can do unbiased gene expression clustering. So all of this can be done with Loop Browser. But of course, there are things that Loop Browser doesn't do. And in those cases, there are a lot of really nice community tools that are available out there in the world for doing additional analysis. And this is something that we're familiar with, and we're happy to point you in the right direction. Typically, they fall into R-based and Python-based approaches, although there is some intermingling, and there are some standardizations that are starting to happen. But generally, this is what we're looking at, and there are some really nice tools out there. So that's where I'll leave you for now, and I'm happy to come back and talk a little bit more about data analysis and what that looks like at some point if that would be interesting to the community. But hopefully this gives you a good idea of what Visium is, what we're thinking about in Spatial, and what you can do. And I just want to say thank you very much for coming and your attention.
Thank you, Jeff. This was wonderful. Yeah, I just want to kind of add with new technologies and, especially with something like pilot chromos and things like that, there are two ways you can think about it. Is either you already have a biological question, and with these tools we can just discuss how you would want to answer it. On the other hand, is like you're just really want to put it in a future grant and use the technology, and we can also discuss what kinds of questions you can answer, because there's a lot of ways we can tweak the assay and have very, very individualized approach.
Absolutely. So before we take a couple questions, I'm going to ask a couple questions. Is is this helpful? Like, did people feel like having vendors come and, have particularly 10x come back? Would you like to learn a little bit more about analysis and hear a little bit more about analysis? All right, I'm seeing some head nods. I think the other thing I meant to mention before is, in the last couple of years, Ruben, Jennifer Bean, and myself had organized a spatial biology seminar series, and so we've kind of pushed that off to Roxy this year, and so Roxy's been working to kind of backfill, see who's presented in the past, and is starting to reach out to local labs. If you're interested in that, we'd also be very happy to expand that and give more opportunity, particularly as you start to generate new spatial data. Working group, if you will. Like, we've had it as a seminar series. We're thinking about how to make it a little bit more interactive. You can come and give a short, this is my question, or this is my challenge. Are people interested in that kind of a forum as well? Maybe a little. All right, so we'll expect some communication. If you signed up for today, Alyssa's going to share the emails so that we can kind of let you know what's coming, and then based on the size and the interest, we'll figure out where to house it. But we're hoping to not only foster the generation of data and work with you as a service corps, but really also continue to expand and engage around spatial biology here. But maybe with that, go ahead.
Yeah, the quick question. So thank you and wonderful presentation. For the spatial biology course, so we also have a single cell sequencing called Yuri is here. So should we go to you or go to Yuri? We're going to use this advance.
Yeah, so if you're wanting to do spatial biology, you should come to us. You can always talk to Yuri. I encourage everyone to talk to Yuri as often as they'd like. But if you're looking to navigate through the spatial, certainly come to us. So I have my email up at one point, so spatial@bu.edu is our new email address. We should have a website hopefully up in the next couple of weeks that'll help you navigate the pricing for both technologies I initially talked about. But yeah, ultimately you'll schedule a time with us. We'll help you navigate what's the starting steps and when you're ready to get going and things like that.
Okay, today's slide presentation will be available for people registered.
Yeah, absolutely. We've recorded it, and so you can go back and watch it, or you want to just look at the slides, or you want to share it with your trainee to watch it after the fact. Yeah, no, that'll be available. We'll make that available both on our side, but I think we'll work with Alyssa to make sure it gets sent out as well.
Thank you.
Any other questions? Yeah, go ahead.
Can you provide more details about how you use cryolysis to transfer the tissue from glass slides to the Visium slides?
Yeah, I guess, in terms of more details, so you'll put your tissue slide in the device in the lid and align it, right? There's an alignment process. You put the Visium slide in the base, you add a transfer buffer to the Visium slide, and then you close the lid, and then you run the program. The program lasts about 40 minutes or so, depending on which version of the assay you're using. And what's happening is that the tissue is being heated up, right? So there's a thermal component to this, and then the transcripts are coming out of the tissue onto the Visium slide during this process.
So it's basically cover onto the slide.
That's right. Yep.
Thank you for your presentation. So I know with some spatial cores, your tissue really needs to be like in a very specific area on the slide, like they give very specific dimensions. Is that true for this as well, or can it be anywhere on this slide?
Yeah, so if you're talking specifically about the Visium technology, there are some guidelines of how to center it. There is some XY flexibility, the actual microscope side. So again, as Jeff mentioned, but I just want to make clear, all you need to do is be able to section your tissue onto a slide. Yes, we do want you to center it to the best of your ability. There's not much wiggle room in the X direction, but in the Y direction, you can move your slide up and down, you can flip it over. So yeah, you will want to centralize it. For the imaging mass cytometry technology, a little bit less, because you're going to put it in and you're going to pick where exactly you're going to lay, for example.
I'd also just add for the Visium technology that we have some really great resources on the 10x website to help you find what Sarah's describing, which we call the allowable area. It's the part that's compatible with the CISIS. So we've got a great document that you can print and use as a template, and we've got a great video, and you can always reach out to me or Roxy and we can sort of walk you through how that looks.
Thank you.
Yeah, Absolutely. 10x has a lot of documentation across all of their different approaches. You're like, well, I do this with, I fix my tissue and then I freeze it, or I freeze it only, or I fix it only and I embed it in paraffin. There are navigations for anywhere from not the collection of your sample, but basically anything post the collection of your sample right up through the data analysis. And so you kind of, for each of the assays, really nicely worked out, kind of click through each of the steps. But yeah, as Shoni mentioned, 10x is happy to help navigate you. It may help, encourage you to reach out to us, and we'll help you navigate here, how to go about that process, depending on what your lab is able to do within the lab versus what you'd like us to help you navigate.
We got an email from GSI that we need to purchase the Vism HD by 26th of this month. So it's a bit too short. It's possible to postpone the deadline, so give us a little bit more time to think about, so we can digest the content.
Yeah, so it's just to be clear, to get that promotion's running through the 26th. I think we're going to continue to work with Elisa around new promotions that are going to come out next quarter. It may not be the same promotion, so if you're really excited and ready, we're still going to keep that promotion to the 26th, but expect there'll be additional promotions to hopefully help other investigators navigate. It may not be the 50%, it may be less, we'll have to talk with and see what 10x is willing to do. The goal is for this to help, get business in, and so, if we don't have enough investigators able to whether participate today or know about it, we're hoping to offer additional opportunities in the next quarter as well.
Yeah, definitely. I encourage you to speak with Sarah and, whatever the timeline you can work. I mean, there are potential exceptions if you're, really set to decide, but if you need more time, Sarah mentioned like in Q4, probably around December, end of the year, it's like very common, but yeah, we'll know and share the details when they're available.
Yes, and we'd like to thank our GSI collaborators to help push the message out, and Yuri for sending this out. But yeah, let us know if you're not getting messages, you found out about this through somebody else, let us know, and we're going to try to include folks that participated today and continue to participate in our events to make sure you get the information out, as well as kind of advertising through the Department of Medicine. Excellent. All right, well, like I said, reach out. Thank you so much. If you didn't get one, thank you guys so much.
Yeah, you bet. Thank you. This was awesome.
Thank you very much. I want to Hi, how are you?
Hey, how are you? Good to see you. Thanks for coming in person. I saw you back there. Yeah, this is wonderful. Off-campus visit. I know. I'm going to
Thank you very much.
That's all I know. So far, I'll keep you posted. Yeah. Oh, thank you. I know you're all okay.